nice to see that we have a good retention rate today. As I mentioned yesterday, the format of today is such that we will basically be covering a few different areas of analysis for single-cell analysis, single-cell RNA-seq. So once you've sort of processed your data, normalized it, and removed all the confounders, and you have your expression matrix that you're happy with. This will presumably only take up until lunchtime. So after lunch, as I mentioned, we have a much more open structure to this course. So we're going to give you 3 options. Number 1 is to, if you have your own problem that you would like to have a consultation on, we're happy to help you out with that. Number 2 is that we have some advanced exercises, which are sort of more open-ended questions that you can work on, and we can try to help you out with that. And number 3 is that you can go home and enjoy the nice autumn weather. So can we have a show of hands? How many are planning on doing number 1? Two. How many are planning on doing number 2? A few more. And the rest of you are planning on going home. You don't have to confess. That's okay. Okay, so what I should mention about the different methods that we're going to be covering today is that we're well aware of the fact that for most of these different types of problems, there are several other additional methods available out there, in particular if you look at bioRxiv. We have really had we had a few guiding principles in choosing which methods which we're going to be covering. Perhaps the most important one is that we'd like them to be available as R packages. Most of them are, but not all of them. Number 2 is that we want them to be reasonably fast so that we can sort of run them in a relatively short time in this classroom setting. And then we should, of course, also confess to the fact that we do have a certain bias for the methods that sort of some of them we have developed and others our friends have developed. So we tend to look more favorably upon those methods. So please keep that in mind, that there is a certain editorial bias in the choice here. So with that, I will hand it over to Vladimir to explain about clustering.
Hello, can you hear me? Yeah. So, yeah, we actually left this chapter dealing with confounders with the read table for the exercise yesterday night, if you remember. Do you have any questions about this? It's basically the same principle that Davis showed you with the UMIs. Okay, so then I'll start with clustering. So clustering is quite important questions with single-cell RNA-seq data. And but you can only do it after you've done all the steps that we covered yesterday. So you do the mapping, quality control, normalization, finding confounders. And after that, when you're sure that you removed all the technical variation from your data, that's the stage where you can start doing some downstream analysis like clustering. And clustering for single-cell data is important because in contrast to the bulk RNA-seq data, here you have many more samples. And basically what you want to see sometimes is what's the structure of your data. Do some cells cluster together in one group, or the distribution is completely homogeneous and there are no clusters in your data. So here I'm going to talk mostly about unsupervised clustering. So so we need to identify groups of cells based on the similarities of transcriptomes without any prior knowledge of the labels. So we have no idea what the experiment was, and we just want to try to identify the cell labels, put them in different groups. Sometimes you actually know what was your experiment, so you expect the clustering, some kind of clustering in your data. But in many cases, for example, in the new large Drugbased data sets, when you have 45,000 cells, for example, it's very hard to predict what the structure of your data. So the only way to do it is to cluster it. So usually, clustering is a very hard problem. It's very computationally expensive. And the first step is usually to reduce dimensions of your data. So yesterday we covered a couple of the approaches, how to reduce dimensions. So one of them is the PCA. So in PCA, you transform your matrix, which has several cells and a lot of genes. So what you do, you reduce the dimensions of the genes. So instead of having thousands of genes, you reduce it to some small dimension. Also, there is a TsKnee that I covered yesterday. This is another approach where you embed your data into 2 dimensions. And then basically what you have is this 2D map. And what you can do, you can apply some density clustering on this map, trying to find the density, the higher density of points on the plot. Another thing is the spectral decomposition. So basically, in this case, it's similar to PCA as well. So you find the spectrum of the matrix, basically find the eigenvectors, and then you work, instead of using all the dimensions again, you only take the first few, and you apply clustering to the first few eigenvectors of your matrix. So I think this is one of the most the necessary step, because the data sets are getting larger. Well, I mean, for most of you probably, I don't know. So how many cells do you analyze? Less than 1,000 cells or more than 1,000 cells? Who analyzes more than 1,000 cells? 2 people here. So all the 3. So all others analyze less than 1,000 cells, right? So for less than 1,000 cells, I think it's not a big deal. But for more than 1,000, more than 10,000, you get into trouble. So you'll have to spend a lot of time for clustering if you don't do dimensionality reduction. So, so after you reduced your dimensions, you can actually apply the clustering algorithms. And here I'm going to go through some of them. The first one is the most popular one, is hierarchical clustering. You probably all know about it. So basically, you have your raw data, but then what you do, you basically find similarities between pairs of cells and put them into one group, creating the first level of hierarchy, and then you go further and create the second level of hierarchy, and so on, until you get to the group where you have all your cells. So this is, I mean, quite efficient approach for small data set, and it works fine. Another clustering method is Kmeans. Kmeans is also very popular in all different kind of research, not just in single cell data analysis. So what happens in Kmeans, you have your cells, for example, and what you do in an iterative manner, you find basically some kind of cluster centers. You start with random cluster centers, and then you compute similarities between the cells again and assign cells to these cluster centers, making this kind of clusters. And then basically, in an iterative manner, you refine the cluster centers. So based on this group, you can find a new cluster center, which will be different from this. And then you do it iteratively until your clustering converges to some stable solution. Yes, so that's another type of clustering. Another also very popular type of clustering is graph-based. So again, your cells are represented in a graph. So where basically the nodes are the cells. And then what you want to do, again, you want to find groups of cells which are closer together in this graph. So these approaches are quite efficient. So sometimes they can be very fast. So they are favorable for large data sets. So we cover one of the methods which uses this approach. So what are the challenges of the clustering? So first of all, as I said, this scalability is a major problem, because in the last 2 years, the number of cells in the experiments grew up to 10 to the 4, 10 to the 5. Another problem is when we do unsupervised clustering is that we don't know the number of clusters. And most of the algorithms actually require you to define the number of clusters in advance. For example, before running Kmeans, you have to say, I want to find 3 clusters in my data. So basically, it will find 3 clusters for you. Some algorithms can predict the number of clusters. Some algorithms do not require the number of clusters. Like, for example, density clustering, it looks at the data and it finds the clusters automatically. But again, some of them actually require you to provide the number of clusters k. And another thing is tools are not user-friendly, so I'm not sure about this anymore. That was a couple of months ago. So now there are quite a lot of tools. Whether they're friendly or not, I think you have to decide, but some of them are really tough to run. So I'm going to cover several tools that are available at the moment. The first one is Sincera. It's actually, the clustering approach here is quite simple. So it's based on hierarchical clustering, simple hierarchical clustering. There is some pre-processing steps. Also, it has the ability to predict the number of clusters. And I'll show you how it does it afterwards. The second algorithm is called PCA Reduce. That was developed in Oxford. And basically, this algorithm combines PCA and Kmeans in iterative hierarchical clustering. The next algorithm is SC3. This was developed by us. And again, here we use Kmeans plus dimensionality reductions, and also additional step is consensus clustering, where multiple solutions are combined together into a single solution. So these two approaches, TsKnee plus Kmeans. So as I said, in TsKnee, you project your data into 2 dimensions, and then you can use either Kmeans, as we do here. So this is actually the approach which was used in some of the papers. So we just took it and tried. And Seurat package actually has had a similar approach. So it also computed the TsKnee map, but then it applied the density clustering instead of Kmeans. However, Seurat has been updated recently. So in the latest version, they no longer use TsKnee map for clustering. So now they use a graph approach similar to the approach which was used in this paper. You can have a look. So I haven't tried that yet. So what I'm going to cover today is the previous version of Seurat. But if you have questions, we can have a look at the new one. And the last one is SNM Click. This is also a graph-based method. So you basically construct a graph from your data, and then you try to identify groups of cells in your graph. So that's basically it for the clustering introduction. In the next chapter, I'm going to show you how these methods work on a data set, on the real data set. Do you have any questions so far? Yes?
In the SC3 part, there is a step called a consensus.
Yes.
So, did you usually do it in the TsKnee plus Kmeans method, whatever, to determine what the cache should be before you
So the question is, in SE3 tool, there is a consensus step. And the question is, how do we identify K? So in SE3, we don't identify K. We basically provide, we require the algorithm to cluster for the K clusters. So this is an input parameter that you have to provide to the tool. Instead, what we do is basically you can provide a range of Ks, and then you can look at the range of Ks and see how your solution changes from one K to another, and whether you get a better structure using a different K. Yeah, we do. We also do have a method for K prediction, and so the common pipeline would be you first run the prediction algorithm. You find it gives you 1K, and then you basically play around this K. So you say, I want to look at the clusters from K minus 2 to K plus 2. I'll show you how it works later, yes. So any of you used any of these tools? No? You haven't? Yes? What's your?
If our algorithm has capability to predict the number of K, then is there any trade-off between linearity for the algorithm?
So the question is, when you predict the number of Ks, is there any trade-off, or is there any disadvantage of using prediction algorithm? I think it depends on the, I mean, every algorithm is different, so you have to look what it does, first of all, and what's behind it. So, and you can trust one of them and not trust the other one. So it depends on your preferences, but all of them are different, and some of them can give you completely different Ks. In this case, you don't know what to do, right? So one of them says there are 2 clusters, the other one says there are 20 clusters. So you have to be, I think the best way is to try different methods and to look whether they give you similar results or not. The same question? Okay. Yeah, so usually the best way is to run different methods and to compare the solutions of the methods.
And so the other thing to say is that there may actually be sort of multiple good answers to the question of how many clusters there are, right? Because it might depend on sort of what level of granularity that you're interested in. So if you're interested in a more coarse-grained subdivision, then say some low value might be more appropriate. But for someone who's more interested in a more fine-grained subdivision, then a significantly higher value would be appropriate. So it really depends on sort of what level of resolution that you're interested in, and in some cases both answers could be considered correct.
Yeah, so there are multiple data sets where you have several hierarchies in your data set. So you have, for example, 4 large cell types, and then inside of each cell type you have some sub-cell types. And basically both answers would be correct in this case.
How computationally, how long does it take to run if you've got, say, 500 cells?
Depends on the tool, again. So
In terms of SE3,
SE3, 500 cells is fast. Yes. So limitation of SE3, if you're interested, at the moment we go up to 5,000 cells. So if you tweak one of the parameters, you can run 5,000 cells in about 20 minutes. But we don't recommend to run it for more than 5,000 cells. And whereas, for example, Seurat at the moment can process much larger data sets. But it's actually, we tried it on a smaller data sets, and the results are not as accurate as with SE3. So I think depending on the size of your data set, on the type of your data set, probably it's better to find out which tool works best for this kind of data. So we're going to use, in the next chapter, we're going to use a data set which is pretty small. It's only I think it only consists of 50-something cells. And so this data set is a real data set. It's a patient data set. And so we have the patient with this disease, myeloproliferative neoplasm. And it's known that this disease is highly heterogeneous, and there can be multiple subclones coexisting in the same patient at the same time. So what we saw in this patient, using the Sanger sequencing genotyping, that basically there are two loci. Sorry, there are three different subclones: wild type, and TET2 mutant, and JAK2 TET2 mutant. So basically, we had this a priori knowledge that there are three subclones in this data set, and we thought whether are we able to identify three clusters in this data using this prior knowledge or not. So, but originally, I mean, the question was, we just wanted to cluster the data set. So this genotyping knowledge is not, we didn't use it in a way that it was in a supervised manner. So the first approach was just to cluster the data to see what's the best solution, how many clusters we get, and then basically look whether our clusters correspond to the subclones identified by Sanger sequencing. So here, yeah, so the data set has 51 cells and about 9,000 genes. And this is already quality control data set and normalized. So we used RUVG here. There was ERCCs in this data set originally. So this is a filtered, good normalized data set. So we'll start with SC3. So if you run all the commands from the top of this page, then if you load the data set using this command, and then we want to run SC3. So SC3, originally, it was only interactive tool. So you would run it on your data matrix, and it would open in a web browser, and then you would be able to look at different solutions. So we modified it, and now, because of the scatter, I made this scatter integration. So now SC3 can take the scatter object, and you can work with the scatter object directly. So you don't need to create any new data matrix and so on. So basically, we try to utilize the convenience of the scatter. So because we don't have a scatter set for this particular data set, so we need to create a scatter object from the data set. And SC3 provides this function, create SC set, and then you give it the data matrix that you have, and basically it will create a scatter set for you. And then, so we also want to run the calculate QC matrix just to be able to use all the scatter parameters in our plots to be able to visualize them. And then basically we need to run this SC3 prepare function and also SC3 estimate K. So this is the function that SC3 allows you to calculate the optimal number of clusters. So SC3 actually tells you that it thinks that there are 3 clusters here. And once you've done this, you are actually ready to run SC3. So as I said, the optimal number of clusters is 3. That's what SC3 tells you. But if you don't believe it, you can actually control the number of clusters in this function. So I define the number of clusters. I want to run SC3 from 2 to 5 clusters because I know that 3 is a good number. But if you don't like it, you can change this parameter. So this function will run SC3 for you. And then for the convenience, again, we're going to use SC3 interactive function, which will open your result in a web browser in an interactive way. And then you will be able to see how your results look like. But also there are all other functions available where you can create the same plots that you see in the interactive session and in your normal R session. So interactive is an optional thing at the moment. So could you please run all this code and then start the interactive session, and then we can continue. I'm going to do the same here. Yeah, I will.
Could I?
Yeah.
I saw a step of removing duplicated
Previous chapter?
Oh, yeah. If you go back, I saw the removing duplicated genes.
Yeah.
So in micro-reflectome, there was some same gene from different proof. I thought that is the meaning of duplicate. So in this context, what's the meaning of duplication?
So here, yeah, so the question is, why do we remove duplicated genes? So I think this filter is, at some point, I had a data set where some genes were duplicated, and basically it screws the algorithm because you actually have the same identical rows in your matrix, which makes it, basically there was some error when I run clustering algorithm. So basically yes, and then I just remove those rows because of this. So this is, I think this is a sanity check. It's more like a sanity check. You shouldn't have duplicated genes in your matrix in the first place, right? So if you run all the commands, you should be able to see similar picture in your browser. So do you see this? Okay, everyone? Okay, so this is the interactive session of SC3 which shows you all the results computed from the data sets, from your data set, sorry. So here we have some parameters. So again, we asked it to cluster from 2 to 5 clusters. Also, this panel shows you different data from the scatter object, right? If you remember, yesterday we used some of them to color code the PCA plots, right? Percentage of ERCCs, percentage of total features, and so on. So these are exported from the scatter object, and we can actually also visualize them on all the heat maps in SC3. And then there are several panels here on top which help you to find out if your clustering solution is good. So let's have a look first. You can actually explore what's happening here. So if you follow the questions on this page, so the first exercise is explore different clustering solutions from K from 2 to 5. And also try different checkboxes on the left side. So the first what you see is the consensus matrix. As I said, in SC3 there is a consensus step. So what happens, SC3 applies different distance metrics, different dimensionality reductions on your data. And then it gets multiple different solutions from these different pipelines. And at the end, because all these solutions may be similar, but they are slightly different, what we wanted to do is to combine them together using consensus approach. And once you combine them, this is your final result. So the final result is a consensus matrix. And the idea behind the consensus matrix is that on the blocks, on the diagonal, you actually have your clusters. All the diagonal elements are not your clusters. And what we want to see is that we want to have as much as possible red color on the diagonal and as much as possible blue color off the diagonal. So, for example, if we change k from 2 to 5, we can start with 2. So 2 clusters, you basically what you see is that, yes, you have red color on the diagonal, but the blue color of the diagonal is not blue enough. So it's not a good clustering solution. And the color here corresponds to the agreement between multiple clustering solutions. So when the color is red, it says that different multiple solutions actually think that these 2 cells belong to the same cluster. We have cells in columns and cells in rows. So if you take this cell and this cell, you find the intersect. And the red color means multiple clustering solutions think that these 2 cells belong to the same cluster. So if we move to 3 clusters, now we can actually see a nice structure. So you have 3 clusters, and off-diagonal elements are quite blue, which means that multiple different clustering solutions think that these 2 cells do not belong to the same cluster. So 3 clusters is a good structure here. Once we move to 4, what we can see is probably the first cluster, which was the largest one, is now split in 2 clusters. But here we can see that this element is not blue enough. So probably these cells are still very similar to this cell. So probably this is a one big cluster. And if we move to 5, the same thing happened here. So we got some cluster of cells which are probably outliers because they don't look like they belong to. They are very similar to this cluster, but still they are very different from each other as well. So we thought that this interactive approach to visualizing this clustering solution in this manner is kind of useful. So we find that it actually helps you to understand what k is good and whether you need to increase it or decrease it, and it's kind of very helpful. So as I mentioned, you can also on top of this heat map, you can add different variables from the scatter object. So let's see if we check this checkbox and this checkbox. So what we can see now is that the top 2 rows now show us the log 10 of total features and the log 10 of total counts. And this is exported again from your scatter object. So this is calculated by scatter. And I think it's nice to visualize it because actually you can see sometimes that your clustering is biased. As I said, if you don't normalize your data set, for example, and apply clustering, what can happen is that one cluster can be separated from the rest because of the library size. So you will be able to see whether your library size in this cluster is higher than in any other cluster. And then this is not good because the clustering is basically biased because of the library size. So you can actually explore different parameters and to make sure that your clustering is reasonable.
Just ask, how's
Yeah.
the percent dropout? How's that calculated? Is what you see in this data set typical?
So the percent dropout is calculated, as I said, it's calculated by scatter when you create scatter object, and then you run calculate QC matrix function. I think it's, is it just, yeah, I think it's just the percentage of dropouts in every row of your matrix, maybe averaged. I'm not sure. So Davis probably knows the details more. But I think, I mean, let's see how it looks like. Yeah.
So do you reckon that here there are 3 clusters more than 4?
Yes, we think so. Yeah. So this is the percent of dropout. Well, basically, yeah, you have it per cell, right? So it calculates the number of zeros you have in your column and divided by the total number of genes you have in your, yeah. Okay, so additionally, as I said, we have some other multiple tabs here. The first one is a silhouette plot. Silhouette plot also helps you to find out whether the block diagonal structure of your matrix is good. For example, and what's important is this average silhouette width. So if we change K from 2, so when we ask for 2 clusters, silhouette width is 0.62. So if we increase the number of clusters, for 3 it's 0.74. And then with 4 it goes down again. And with 5 it goes down again. So it's similar to the consensus matrix, but instead of looking at the matrix with your eyes, this is kind of quantitative measure of block diagonality of your matrix. Also, SC3 provides cell labels. So these are the cell indexes from your original data set in the order that you basically, in the order of the cells in your data set. And what's nice about this is that, so we're asking for 5 clusters. It shows us 5 clusters and cell labels in these 5 clusters. But also they are colored with different colors. And the color corresponds to the number of clusters minus 1 from what you have at the moment. So we have 5 clusters, but we have 4 colors. And 4 colors corresponds to K equals 4, right? And this is sometimes very useful because what we can see here is that these 2 clusters, if you ask for K equals 4, what you get is you would get this, this, and these 2 clusters would merge together, right? So when you move from 4 to 5, this cluster is split in 2. So sometimes it's useful to see what happens when you change K from K to K plus 1 to see which clusters actually split. So if we play with it, for example, now I move it to 4. So you see that this guy splits in 2 when we move back to 5, right? So there is also stability tab here. Stability also, it only works when you have multiple Ks here. So it calculates stability of each cluster. So now we have 5 clusters, and it finds out when you change your K from 5 to any other number here, whether this cluster stays the same or not, right? So if you have multiple Ks and you change K from 2 to 5, so some clusters cannot change, and it will always stay the same. Some clusters can split, some clusters can merge, but some can stay. And basically stability is, if it's 1, it means that this cluster is very stable. It will not change if you change K from 2 to 5. So this is, the first 4 tabs are helpful to validate your clustering and to find out what K is the optimal one. And the next few tabs are basically visualization of your data. So this is an expression matrix. So basically it's just the heat map of cells in columns, genes in rows. Genes are clustered in 100 clusters for convenience because sometimes the number of genes is very large. And again, you can visualize these different scatter metrics in the same heat map. And then you can actually see, looking at the expression, whether your clustering was good or not. So you can also validate your clustering using this heat map. So if we change K from 2 to 5, well, with this data set, it's really hard to say whether 3 is good, 3 is better than others. But, you know, for example, here you can see that these genes are expressed a bit higher than this, right? So probably it's an okay solution for this data set. We also provide TsKnee map, which is, I mean, here we just follow the popularity of TsKnee because everybody likes TsKnee. So I'm not sure whether this is useful, but again, it just projects your data in these 2 dimensions, and it also color code the cells with the, so the color correspond to the clusters identified by SC3.
Sorry, what does the perplexity does in this?
Yeah, so this is the parameter I explained yesterday. So perplexity is one of the parameters of the TsKnee transformation, and I think it corresponds to the what kind of density you want to have in your projected data. So it basically changes the density of the points on your map. So if we change it to 7, I think it becomes a bit more homogeneous. Well, yeah. So,
your range factor is small.
Yeah. So this range is actually the default range. I don't remember how we calculated, but I think for smaller data sets, TsKnee is usually always homogeneous. So unless you have really strong clusters in your data, it will always look like that. With larger data sets, it works better. So I wouldn't recommend use TsKnee for smaller data sets.
How many cells do you need?
Large, well, more than 1,000 I think is good for TsKnee. More than 500 maybe. Several hundred is good. Yeah.
So I also remember yesterday you mentioned that you get different results if you run it multiple times.
Yes.
So is it recommended then?
TsKnee.
Yeah.
Well, as I said, you can actually, yes, you're right. So if you run it multiple times, you will get multiple results. But if you're, as I said, if your clusters are stable and strong, the results would be, I mean, they will show the same thing. It will be, the map will be a bit different, but still the clusters will be the same. If your data is not strong enough, so the signal is not strong, then it will be probably very, you know, it might change the structure. But also there are several ways, like in Surat, you can increase the number of steps, because TsKnee, I think it's also iterative, so you can increase the number of steps that you run it for to wait until it converges to some, you know, stable solution. But I had a bad experience with TsKnee, so I wouldn't recommend it to anyone. This tab is included because of the popularity of TsKnee. And the last three tabs are basically biological analysis of your data. So the first tab is differentially expressed genes. So it's basically how in SC3 we define differentially expressed genes as in a very simple way. So if the gene is expressed, if the difference between expression of any gene in one cluster is different to the expression in any other cluster, then we define the gene as differentially expressed. So for example, it's not expressed in these two clusters, but it is expressed in this cluster, then we define it as differentially expressed. This guy is different, so it is expressed in this, but less expressed in the third one. It's also kind of differentially expressed. That's how we define differential expression. So actually, SC3 calculates all these values for all case in advance, so you don't have to wait until it calculates it for different case. The second tab is marker genes. So marker genes is similar to differentially expressed, but now we require the gene to be only expressed in one cluster, right? So if the gene is mostly expressed in one cluster, then this is a marker gene, and it's not expressed in any other cluster. So what SC3 provides you with this. So here you have your clusters. Where is it? It's not here anymore. So yeah, so you have cluster 1, 2, 3, and these are the marker genes for cluster 1, these are the marker genes for cluster 2, and these are the marker genes for cluster 3. You can also run gene ontology analysis. We're using gprofiler here. So you can choose the cluster that you're interested in. For example, you want to run gene ontology on these genes. But yeah, so we didn't install this. You will need to install it on this R-Selenium package on your computer to be able to run it. But what it does, it only exports your gene list to gprofiler. So it opens it in a separate web page, and then you can run it in gprofiler. So it's quite convenient when you don't want to copy-paste the gene names. Yes?
So in previous stuff, in the differential expression, how exactly does it work? So what differential express? Because there is no separation with the classes.
Yeah, so the question is, what's the difference between differentially expressed genes and marker genes? So differentially expressed genes will look at least one difference between any two clusters for a given gene, right? So, for example, this gene is expressed in these two clusters, but less expressed in the third cluster. And that's how we define differential expression. If there is at least one difference between any two clusters, this gene is differentially expressed. Whereas marker genes, we require the gene to be expressed only in one cluster and not expressed or less expressed in the other clusters. Yeah, so actually there is an explanation of how the algorithm that we use to define the genes, and also the paper is on bioRxiv. You can find all the details there. And
in this context, the meaning of differentially expressed genes may sound like different from upregulated genes or downregulated genes.
Can you repeat the question again?
So in this context, the meaning of differentially expressed gene may be different from upregulated gene or downregulated.
Yeah, so the question is, the meaning of differentially expressed genes in this context is different from upregulating genes or downregulating genes. Yes, this is true. That's how we define differential expression ourselves. So it's, I mean, the classical differential expression, when you have two groups, you compare two groups and you find out which genes are upregulated in one group or downregulated in one group. Here we have multiple groups, so differential expression contexts change. And we define it in a way that if there is at least one difference between any two clusters, then the gene is differentially expressed. So I think so we're gonna cover differential expression in the next chapters, and I think for most of the data sets, single cell data sets, again the differential expression tools require you to use two groups. So now you have multiple cells in each group compared to the bulk data set when you can have like 2 replicates or 3 replicates. Now you have hundreds of replicates, right? But they still require you to use 2 groups. So here, because we didn't want to do pairwise comparisons between the groups, that was the easiest way to define differential expression. So it's different from the context of classical differential expression. Yes. And the last tab is the outliers. So it's also a simple approach, but I think it's kind of interesting. So here you can see your cells again from 0 to 51, and the clusters are colored. And what we look at, we actually look at every cluster separately, and in each cluster we try to find cells which are the furthest away from the center of mass of the cluster. And then we say that these cells might be outliers. So because the clustering, Kmeans doesn't care about outliers, it will basically put all the cells in some cluster anyway. So, but those cells can be actually very different from other cells in this cluster. So this is the way of looking at the outlier cells. And sometimes actually it's very helpful. So you have a cluster and then you find out that some cells are different. You look at the expression, they are different. So for example here, it says that so I think this is the strongest one, right? So it's cell number 4 from the right. So if you look at the expression matrix, yeah, it's not very different. So I think this cell, for example, the second cell in cluster 2, oh, it was not identified. Yeah, so it's it's probably not a good example. But we found several data sets where this actually helped us to find. So these cells, I think, are not very different from each other. That's why it's just, yeah.
Outlier score. Does outlier score mean the distance from the centroid?
Yeah.
But why? I expect some gradual distribution of
Yeah. So the question is, what does the outlier score mean, and why it is not? Why is it discrete? So we have zeros for some cells and some positive values for other cells. So the outlier score shows you how far your cell is from the center of mass of the cluster, as I mentioned before. But also we put a threshold on the outlier score. So these cells, they also have some outlier score, but it's below the threshold. That's why we make it 0. So here I think we define, this is the threshold that we use, very strict threshold. And we only show cells which are above this threshold, and all other cells are 0.
Is it possible to adjust
Yes, so you can control this parameter. So if you reduce this, you will see other cells that will have some other values which will be not 0, but they will be lower than these ones. Yeah.
Would you remove that outliers from your graph from the analysis?
If you look at that in the last graphic, the two cells that go to the markers, those cells with high, I'm guessing they have very high total feature count. This is different.
Yeah, so the question is, would we remove outliers from the further analysis, right? So in this case, I wouldn't, because I looked at the expression and it doesn't look very different. So, and also, SC3 was able to find marker genes for us and differentially expressed genes using these cells, right? So as I said, we saw several cases where the cells, like a group of cells, was completely different from the rest of the cells in the same cluster. And it turned out that these cells were sequenced using the different protocol. And that's why actually they were from the same stage of the development, but they were sequenced using the different protocol, and they were defined as outliers. So I think you need to look again at different metrics here. So you need to look at the expression, how the expression looks like for these cells, whether they are consistent with the marker genes. For example, if your outlier cells have the same expression in the marker genes as all the other cells in the same cluster. So I think, you know, it's kind of subjective opinion here. You need to.
Yeah, I mean, I think really sort of one of the features of K-means clustering, right, is that if you ask for 5 clusters, you will always get 5 clusters no matter what, right? And these methods are known to have problems with sort of rare outliers and sort of small groups. And this is sort of a way to help you identify those. So I think in many cases, what the sort of this method calls as outliers are probably sort of quite benign, and they're not nothing to be alarmed about. But the reason why we have this feature is really to sort of to give you an idea, help you figure out which cells are the ones that you should look at and sort of keep an eye on, as they are the ones that don't fit the mold really well. But as I said, we don't really know, but it helps you narrow down which ones to inspect, really. That's the purpose.
So I think that's it about SC3. So you can actually, as I said, you can create all these plots in your R session. You don't need to open the interactive session at all. So if you're interested, have a look at SN bioconductor, and all the functions are listed in the vignette. So I actually tried to show how to plot everything that you saw here in the interactive session. So the best way is probably to run it first, then open the interactive session, look at the solution that you like, and then go back to the session and plot, make all the plots in the R session. Yeah.
This is the latest version of SC3.
This is the latest, yes.
Because in the previous version there is some download buttons.
Yeah, so I removed all of this. Now you can do everything in the non-interactive R session.
But if I want to do further analysis based on the clustering result of SC3, how can I get the clustering information of these cells?
So if you want, the question is, you want to use another tool for clustering?
No, if I want to do further analysis based on the clustering result of SC3.
So the question is how to export the results.
Yeah.
Yeah, so as I said, you find using the interactive session is a good way to find the best solution. Then you go back to non-interactive session, and then you can export all the results. There are functions there to export in Excel, or you can export it in a data file. So if you go to the bioconductor page of SC3, open the vignette, you will find all the comments there. Okay, so let's move on. We've done all the exercises here. So the next tool is PCA Reduce. So the same way as SC3, it doesn't provide quality control normalization. So as I said, you need to do it in advance using Skater, for example. So that's the command that you need to use to run. No, so here basically it requires you to filter the genes first in any way that you like, and log transform the data before running it. So what we're going to use SC3 gene filter, which I didn't mention before, but it kind of removes genes that are lowly expressed, so they're mostly zeros in all the cells, or the genes that are always expressed, like they are not expressed only in few cells. Yep.
This is not needed for SC3?
This is included in SC3, so when you run it, it will do it for you. So here, because it's recommended to filter your metrics, we use the same filter as in SC3, because I think there is no filter provided with PCA Reduce. And basically, I didn't go into details of the PCA Reduce algorithm, but as I mentioned, it's a combination of the PCA and hierarchical clustering together. So what you need to do is you need to run this. So this is our input matrix. Then we need to define these three parameters here. The first one is the number of runs that you want to run it for, because it's also stochastic, so you will get different solutions after running it multiple times. This is the parameter which defines the number of dimensions to start the clustering with. So what it will do, it will basically start with 30 clusters, and then it will decrease the number of clusters at each step, and you will get a hierarchy from 30 to 2. And the last parameter is a method. So there are two methods. One is perform sampling-based merging, and the second method is merging based on largest probability. So if you're interested in the details, you can go to the paper. It's published. I think it's Genome Biology, and find out what's the difference between the methods. Here we're just going to run this one default and see what the solutions are and whether they are different from SC3. yeah, so once you run this function, as I said, you will get 10 solutions. And we also provide this function to merge the solutions together. Just run it, basically, at the moment. And then we want to plot the heat map of the solutions. So this is how it looks like. So in columns here, we have cells, again, 51 cell. In rows, we have clustering solutions, 10. And the color corresponds to the clusterings, right? So we asked, here we ask for 3 clusters. Here we asked for 3 clusters, and that's why we see 3 clusters. So we have 3 colors in every row, right? So this is what you get after running it 10 times. As you can see, there are some similarities between solutions. So these cells, well, most of these cells are always belong to the same cluster. These first cells, excluding these 2, also kind of belong to the same cluster. But the size of the clusters is different from what you saw in SC3, right? So you have a large cluster, sorry, one large cluster and smaller clusters. That's what I was saying about the usability of the tool. So basically, we run it. This is what you get, and it's actually, I don't know, it's hard to say whether the solution is good or not. So that was the purpose of SC3, basically with an interactive session of SC3 to help you understand what's going on with your data. But that's what you get in PCA Reduce. So if you change K to 2, that's what you will see. So again, this is 10 runs of PCA Reduce with K equals 2. So you have 2 clusters in every row. And there are similarities. Again, you have kind of large cluster and one small cluster. And if K equals 5, I think it's again similar. Somehow it finds large cluster and very small clusters as well. This is K equals 5. So yeah, so this is PCA Reduce. SC3 is actually also stochastic because it uses Kmeans. It's also stochastic, but because of the consensus step, actually the stability of the solution is much higher than in PCA Reduce, for example. So we compared stabilities, and SC3 is quite stable, but it's still stochastic. So for this data set, if you run it multiple times, you will always get the same solution. It's quite stable. But there are some data sets where you run SC3 multiple times, and you will also get different solutions. So that's it about PCA Reduce. Now we move on to TsKnee plus Kmeans. So this is very simple. So you first do TsKnee, you get a map, and then you apply Kmeans to these 2 dimensions that you have. So if you run TsKnee on this data set, you already saw it in SC3. That's exactly the same thing that we did in SC3. So here the cells are not colored with their clusters. It's just the TsKnee map of your data set. And now we want to run Kmeans. So if we run Kmeans with K equals 2, Kmeans finds 2 clusters for you. So this is one cluster, this is another cluster. Again, here it all depends on the TsKnee map. So if your TsKnee map has quite distinct clusters, Kmeans will find these clusters for you. This map is quite homogeneous, so I think what it does, it just split it in 2, and one half becomes one cluster, the other half becomes the other cluster. So you can run Kmeans with different Ks again, and it will find different number of clusters for you. Okay, so here we also, because TsKnee is also stochastic, so we can run it again 10 times, right? With K equals 3 and perplexity equals 10. And this is again the solutions that we see. And actually here, the size of the clusters are quite similar to what we saw in SC3. So we had the larger cluster, but 2 clusters of similar size. So I think the solution of TsKnee is actually similar to SC3, but not to PCA Reduce. So this is k equals 2. Again, you can see it's quite consistent. So even though the TsKnee map is quite homogeneous, but the solution, there are some exceptions, but the main cluster is quite consistent. And this is k equals 4. It's become a bit more messy. Okay, so the next tool is SNM Click. SNM Click is actually not fully R package. So there are some bits of code which you have to run in Python. And as I explained before, SNM Click is basically it's a graph-based approach based on nearest neighbor algorithm. So it tries to find nearest neighbor in the graph constructed from your data set. And then from this information, it tries to find the groups of the nearest neighbors and assign them to the same cluster. So this is the code you need to run. It's not straightforward. So there are 3 parameters you have to, 4 parameters. The distance measure you want to use, and k, r, and m parameters. If you want the details again, please look at the paper. Then you need to, this function we provide with our own package. So this package we made for convenience. So you can actually use this function without doing too much. You can have a look how it looks like. And then you I think this constructs the graph. And then the next thing, which is run in Python, finds clusters in the graph. So, and the output that you get is just this, right? So it says that it found 2 clusters in your data set. And also it provides the cluster labels for you. So this algorithm is deterministic. If you run it multiple times, you will get the same solution every time you run it. But it also finds a number of clusters for you, so you don't have to define k yourself. So what it says, it finds 2 clusters, and it provides you with the labels. So any questions about SNM Click? The next one is Sincera. Sincera is it's a big toolbox where one of the things that it can do is clustering, and it's quite simple. So it's just simple hierarchical clustering. It first performs the Z transformation of your data, so it just finds Z scores instead of the genes, and then it computes the correlation, Pearson correlation between the cells. And then it uses hierarchical clustering of this distance measure. Also, it provides an algorithm to predict k, and it predicts k in a way that it generates the hierarchical tree, and then when it finds a cluster with only 1 cell, so it goes from the top of the tree until it finds that there is a cluster with 1 cell. And then it stops and it says, this is your optimal k. And that's how it works. But this can be dangerous sometimes. So if we look at the data, so this is our data set. So this is actually different from what I just said. So if you ask Sincera to cluster our data with k equals 3, this is what you get. You get 2 large clusters, 1 small cluster. Yeah, but if you run the k prediction algorithm, it will tell you that k equals 1. The problem is that at the first step of hierarchy, it will find this cluster with this cell, which is probably an outlier, and it clusters it in a separate cluster, and it reports that k equals 1. So sometimes this algorithm is not very efficient. Any questions about Sincera? Okay, so and the last one is Seurat. So I think this is the most popular one so far. So as I said, Seurat has been updated very recently. I think the beginning of October, and in the previous version it used TsKnee map for clustering. In the newest version, it now uses some graph-based approach. Yeah, so I think they use the same algorithm as was used for site-off data from this paper. And because they didn't provide any benchmarking, I can't really tell you whether it works the same way or it's very different. They just say that they changed it. But however, I'm going to concentrate on the previous version because I didn't have time to update it to the newest one. So in the previous version, the TsKnee map was produced from your data, and then instead of Kmeans, they used density-based clustering approach. So basically, the algorithm tried to find the places on your map where the density of the cells was higher, and that's how the clusters were defined. And this Seurat was used for the larger data set. So if you know the Makosko paper with 45,000 cells, that's where this approach was used. It's quite complicated to run it. So there are a lot of, it's similar to Skater, I think, at the moment. So you define a new class which is called Seurat. The same way as we do in Skater, we define SC as set object. So you also provide it with the data. But I'm not sure if it's as advanced as Skater at the moment. So I think it's a similar concept, but I would prefer to use Skater instead. But you know, a lot of people use Seurat as well. So basically, you create the object, then you set it up using different parameters. You can go to the tutorial. They have tutorials on the website and see what all these parameters mean. Then it computes the variation. You see this kind of plot. Then you run PCA. So I think what it does, it finds the most variable genes first. Then it runs PCA on those genes. And then from the PCA, it tries to find, using this algorithm, it tries to find the most important principal components. And once it finds the most important principal components, it basically creates a TsKnee map based on these components. So the code is not easy to follow, I think. You can just skip it or copy-paste it. And so the final step, as I said, once the TsKnee map is ready, you can run this dbclust algorithm, which is density-based clustering. And so here it's also very sensitive. There is a parameter which defines the density that you want to see in your clusters. So if you change this parameter, you might get completely different number of clusters in your data set. But because this data set is very small, it actually found only one cluster. I tried different density parameters, and it was not able to find. But as I said, I think this algorithm is very good if you have larger data sets, like they applied it to several thousand cells. For smaller data sets, it's not good enough, I think. And we actually, in SC3 paper, which is still on bioRxiv, we compare SC3 to all of these algorithms, and you can see, and we use different data sets, so you can actually see how these algorithms perform. And that's it. I think Do you have any questions about clustering? So clustering is probably one of the first steps that you usually do after the quality control and normalization, because it basically gives you a nice idea what's in your data, what you should expect, and then you can get some ideas and thoughts of what to do further, how to investigate it, and so on. So if you don't have any questions, then are we continuing or we stop?
I think we'll cluster by the coffee machine.
Okay, so now we have a coffee break for 15 minutes, 10 minutes, and then we'll continue.